I'm now pleased to introduce Dr. Christian Abnet to deliver a lecture on gastric cancer in high-risk populations. Dr. Abnet earned his PhD in environmental toxicology from the University of Wisconsin and an MPH in epidemiology. He joined the NCI as a cancer prevention fellow in the Division of Cancer Prevention in 1998. He became a tenure-track investigator in the Nutritional Epidemiology branch of the NCI in 2005. And in 2014, he was awarded scientific tenure by the NIH and appointed acting chief of the Nutritional Epidemiology branch. In 2015, he was named chief of the newly established Metabolic Epidemiology branch. His work focuses on the etiology of esophageal and gastric cancers. He studies the complex pattern of incidence and differing etiologic factors for gastric and esophageal cancers and the multiple risk factors across diverse populations that share high rates of these two cancers. This is a special introduction for me, as Dr. Abnet has been a fantastic collaborator and supporter in my own career development. He's not just a prolific scientist, but he's also a wonderful mentor. He's had the vision to bring together like-minded researchers and to facilitate conversations across continents, embodying the reason why we're all here today. I'm certain that Rachel would have been proud and humbled to have this lecture delivered in her honor. And I'm also pleased to announce that this lectureship will be named the Pearline Humanitarian Award Lecture in perpetuity at all future meetings.
Hello. It's a real honor to give this lecture in honor of Rachel's commitment. I'm going to try to touch on the concept of gastric cancer as a major public health burden around the world. I'll talk a little bit about the burden, some of the work that we've done at the NCI to try to understand where this comes from, and then finish up by looking at some ideas where we may be able to achieve some sort of control of gastric cancer in the future. I had the great fortune of coming into a large upper GI cancer research group at the NCI that had been working for decades before I got there, and it's been a real honor for me. So a lot of the work I'm going to present was done through this large series of collaborations that has worked using resources that were developed by Joe Ferramini, Bill Blott, Phil Taylor, Sandy Dawsey, and others. And so I don't have an acknowledgement slide, but there's dozens and dozens of people who contributed to this work. So as I said, I'll talk a little bit about the burden of gastric cancer that the world faces, the differences between populations, which has been a major theme of my work, the opportunities for gastric cancer control. There's a couple of different ideas: chemoprevention, Helicobacter pylori eradication, or individual level screening. So just to orient people who aren't familiar with these diseases, the esophagus is lined with a squamous cells, and when a cancer forms in the esophagus, typically it's a squamous cell carcinoma, and this is really the most of the world's burden is. More recently, in the United States and Western Europe, esophageal adenocarcinoma has become a more common disease. It's a fascinating story, but it's not really part of what I'm going to talk about today. But then when we look at stomach cancer, we're talking mainly about adenocarcinomas. And there's different ways of subclassifying gastric cancer, but the one that's really important for the etiologic studies that I do is whether or not we're talking about the cardia, which is the top few centimeters of the stomach, or the non-cardia. And the reason that's so important is that this little piece of the stomach is particularly susceptible to developing cancer, and yet there's two completely different things going on. There's gastric cardia cancers that occur in Western countries that seem to be a lot like esophageal adenocarcinomas. But when we look in Asia and in some other parts of the world, this part of the stomach seems to share a lot of risk factors with squamous cell carcinoma of the esophagus, which, although they're adjacent organs, they're different tissue types, and it's really not clear why they share this. But for those of us trying to understand the causes and achieving control of these diseases, it's an important distinction that we keep in mind whenever we're doing work. So the burden of gastric cancer is very high. There's almost 1 million new cases every year, and most of the people who are diagnosed with this disease die from it. In the US, this is considered a rare cancer. It's about 25,000 cases per year. And the fact that this cancer has been seemingly, the rates are coming down, and I'll show you the figures in a few minutes, has led to the fact that there is really not as much research on this disease when you consider that there's 1 million cases a year worldwide. And I think that's because in high-income countries, this cancer is starting to go away. So here is a map from IARC, and so the dark blue are the areas with higher incidence, and the paler colors have lower incidence. You can see that in Asia is where there's a lot of dark blue, and that's in China, and right across Central Asia to Iran and Turkey. And then the other area of high incidence is the Pacific coast of South America. You can see that Africa, for the most part, is pale in color, and sometimes we're concerned that these registration rates are not very accurate, but we think that this is probably accurate. It's sometimes referred to as the African enigma. We have high rates of Helicobacter pylori infection, the main cause of gastric cancer, and yet gastric cancer seems to be a rare disease in this area. So when we look at those country-level maps, it obscures something that there's usually a lot of variation within a country, but for gastric cancer, it's particularly notable. And in fact, when you look at the high rates in China and the very high population, 45% of all gastric cancer occurs in China. So there's close to 400,000 new cases every year in China. So when we work there, we're working on the place where this is the biggest burden in the world. But within China, there's a lot of heterogeneity. This map shows the top 10 percentile and the bottom 10 percentile, if you look at the red and the yellow areas. And this is looking at every county in China. And in 1972, the Chinese government estimated the cancer rates in every county in China. And you can see that there's a real uneven pattern, that much of southern China is not particularly affected by China, but there's areas along the coast near Shanghai and central China and North China where a lot of this disease occurs. And then in these regions, it's truly epidemic rates of gastric cancer. Now, what I can't show you is the difference between cardia and non-cardia cancer using these maps, but I can do an approximation, and that's showing you the esophageal cancer map versus gastric cancer. For reasons we don't understand, wherever there's a lot of squamous cell carcinoma of the esophagus in China, there's a lot of cardia adenocarcinoma as well. And so that map on the left is actually the map of the esophagus, but it very much approximates what we'd see if we had a map of the cardia. Whereas the map on the right is a mixture of cardia and non-cardia. So in the central part of China, there's a mountain range called the Taihang Mountains, and in the esophageal map, you can see there's this large red area, and that represents an area where approximately 10 to 12% of all deaths are due to esophageal cancer. It's the single greatest concentration of cancer anywhere in the world of a single type, and this is followed closely by deaths from esophageal cancer, cardiac cancer. We have a cohort of 32,000 people who we've been following since 1985. Over 3,000 of those people in the general population have developed esophageal cancer. 2,600 have developed gastric cancer. So it's a really unbelievable burden, making it an excellent place to do research and hopefully an excellent place to find some solutions. I just want to mention that as I showed you the map of the world, that in the other end of that Asian stretch of high incidence of gastric cancer is Iran. Again, I'm showing you a map of esophageal cancer because I can't show you a cardiac cancer map because it doesn't exist. But this is the cancer registry from Iran in 1972. The dark black areas in those regions, esophageal cancer and gastric cardia adenocarcinoma are extraordinarily common, almost as common as central China. But as you go along the Caspian Sea, esophageal cancer disappears and non-cardia gastric cancer becomes a dominant disease. After 20 years of work in China, our group decided that we really needed to branch out and we needed to have additional areas where we could do research so we could contrast it with what we were learning in China. And so we've established very productive collaborations in Iran. It's a difficult place for the US government to do research, but it's not impossible. And we have been able to conduct case control studies of gastric and esophageal cancer and assemble a large cohort of 50,000 healthy adults that we've been following now for over 10 years. And we hope to be able to replicate what we've done in China and contrast it with what happens in another population that's genetically and culturally distinct. Following this development, we've also begun our work in East Africa. We've had a lot of great partners that have established their own studies, and we also work a lot with the International Agency for Research on Cancer, and what we're hoping to build is a consortium of studies across East Africa to study both esophageal cancer, and where possible, we will add in gastric cardia cancer. Okay, so the burden of gastric cancer is high, it's unevenly distributed, and that suggests that there must be something going on that's quite different, and we want to understand the etiology of this because that's the best way to think about primary prevention. Once it was clear that cigarette smoking was the leading and the main cause of lung cancer, we had a plan for primary prevention, and that was eliminate smoking tobacco. It's been quite effective in the US. There's still a little ways to go, but we need to think about these other diseases the same way. The rate of gastric cancer has been declining in the US since we first started keeping track. The Connecticut Tumor Registry has records going back to the 1930s, and there's been a really dramatic decline in gastric cancer in the United States. You can see in the most recent SEER update that this continues. You can see that also that the lines between new cases and deaths is essentially parallel. This suggests that we're not getting fewer deaths because of better treatment, we're just getting fewer deaths because there's fewer cases. And this is probably the continuation of the wealth effect. It appears that when countries get more wealthy, gastric cancer seems to go down rapidly, and we think that that's because Helicobacter pylori stops becoming a common infection. Now, these declining rates are pretty much true right across the world. I know you can't read what the colors of all these lines are, but this is Colombia, Canada, the United States, Japan, Israel, Denmark, and a host of other countries. And essentially, we've seen this decline in the incidence rates of gastric cancer for, on the left, men, and on the right, women, just about everywhere in the world where it's being tracked. One thing to note, the yellow line that's from Japan, where they have extraordinarily high rates. And essentially, for a cancer that mostly affects low and middle income countries, it's somewhat fortunate, if I can put it that way, that we have a high income country like Japan that has a very large burden of this disease. Everybody benefits when there's a high income country that has a major problem and they're using a lot of their resources to try to understand the problem. So there's this great effect on the declining rates, but the question about how many people are being affected is a different question. So recently, the IARC made an estimate of what's the future for gastric cancer look like worldwide. As I said, we have just under 1 million new cases per year now. They projected that through 2030, there's two different trends happening. One is that if you just looked at the demographic effect, and this is essentially that this was mentioned this morning, that in many developing countries, economically developing countries, we see that there's an increasingly aging population. What that means is there's more people who are surviving to an age where they develop cancer. The demographic effect would project that we would go from just about 1 million to 1.5 million cases per year of gastric cancer by 2030. But because of this rapidly declining incidence rates, they almost negate each other, and it looks like we should be at about 1 million cases per year for the foreseeable future. That's still a huge burden, and it still means that a lot more work needs to be done. Okay, so I've touched on the burden of gastric cancer. Now I'd like to discuss some of our studies on trying to understand the etiology of these diseases, because if we understand the etiology, we have the chance for primary prevention. So we know that the main cause of non-cardiac gastric cancer is Helicobacter pylori. It's a very common infection. 50 years ago, almost the entire human population was infected with Helicobacter pylori, but if you look in children in the United States now, it's almost completely absent. It seems to go away with better sanitation, better food, all on its own, and then you probably have because the world is awash in antibiotics, you're probably getting a lot of incidental removal of H. pylori with all the different courses of antibiotics that people take. But there's a host of other factors. So when you have almost the entire population affected, and yet only maybe 1% of the world actually goes on to develop a cancer from this Helicobacter pylori infection, clearly there's a lot of other things going on. And that's where our knowledge is much more limited. We know, of course, that age is essentially related to most cancers. We know that the strain of Helicobacter pylori is probably important. There's one particular gene that's present in some strains of HP that makes it more virulent, but we've only scratched the surface on HP genomes. Sex is important. Pretty much everywhere in the world, men are twice as likely to get a gastric cancer as women, and some of that difference goes away after menopause, suggesting that there could be something with sex hormones going on. And diet. Diet is a very popular hypothesis, but dietary studies of cancer are extremely difficult to perform. What we've learned from most case control studies is not well replicated in cohort studies where we start with healthy people. But we think that there are some, and I want to tell you one story about some micronutrients that were actually tested in a trial called the Nutrition Intervention Trial. So for the cardia, and as I said, the cardia is a very important part of the gastric cancer story. The causes differ geographically. In Western countries, we think that the primary cause of cardigastric adenocarcinoma is gastroesophageal reflux disease, just like esophageal adenocarcinoma. And strangely, it seems like Helicobacter pylori infection may actually reduce your risk of developing this disease. And the reason that that may be is that when you have an HP infection, it can cause gastric atrophy. You lose the ability to secrete stomach acid. And now if you reflux, you're refluxing neutral compounds, not acid. And that may actually be reducing the risk of this cancer. In Asian countries, we found that HP causes gastric cancer in the cardia, and that we don't really know why it's that way. It's something that we observed first about 15 years ago, and we've repeated it multiple times. And when we try to assess the worldwide burden of HP-induced disease, this is an important component because of the large number of gastric cancer cases in Asia. As I mentioned, we have this very strange area in the Taihang Mountains in China where we have epidemic rates of both diseases and the very large burden that this places on the local population. And when we're studying these risk factors, we see that gastric cardiac cancer shares a lot of risk factors with squamous cell carcinoma, but not all. For example, we don't think that H. pylori has anything to do with squamous cell carcinoma in the esophagus. This is some of the first evidence that we showed where we were able to show that HP induces cardiac cancer in Asian populations. And at the bottom, we see that for both sites, there's an increased risk for people who test serologically positive for having had, at least at some time, been infected with this bacteria. We recently repeated that in Iran, and we were trying to look in more parts of Asia. Now, when you see these really concentrated areas within China, one of the first hypotheses is that there was a genetic factor that was affecting this, and that maybe we had as a founder effect. These are people living in relatively isolated areas. It's possible that there was some sort of genetic mutation that's very common in these areas. We know that family history is very important risk factor. And for many years, people tried to do a hypothesis-driven genetic testing to understand whether there was a link between the certain genetic variants and risk of gastric cancer. And then the advent of genome-wide association studies allowed us to look at common genetic variants across the genome in a simple, powerful way. And we did this in China a few years ago for squamous cell carcinoma and gastric cancer. And this is a picture. And I just wanted to highlight something. This is a way of looking at using genetic variation that's ancestry informative to understand the population structure. We had brought together some studies that we had done that the National Cancer Institute did in collaboration with folks in China in that high-risk area in central China. We also collaborated with 2 cohorts that are being run by the Vanderbilt University along with the Shanghai Cancer Center, and then a cohort that had been put together a long time ago by Mimi Yu that was Singaporeans of Chinese descent. We put these together, and genomically, they're very different. We can easily tell the different groups apart, and this highlighted the possibility that there may be something special going on in the high-risk area. When we completed the study, we concluded that there was nothing special about those, and that those people are not particularly inbred or something like that, causing something like a founder effect. When we looked across the whole genome, and this is what we call a Manhattan plot where we're looking at each chromosome, we found that on chromosome 10, the strongest signal for susceptibility to these two different cancers were both on chromosome 10, and it wasn't just on the same chromosome, it was in fact identical SNPs. And this is looking closely at 10q23, and what we found is that the strongest signal for either gastric cancer or esophageal cancer was from the set of SNPs in a gene called PLCE1, phospholipase C epsilon 1, and that this was where we thought that the biggest effect was happening. Now, these effects are really modest. They don't create high risk of disease. This is not like BRCA1. This is a very modest increased risk, but we hope that it gives us clues about what could be happening. Now, as I said, we usually separate the cardia and the non-cardia because we don't think that they have the same risk factors. And when we did that here, what we found is that gastric cancer signal from this area of the genome was in fact no signal at all for the non-cardia gastric cancers, but a very strong signal from the cardia. This gene is very interesting. It's been knocked out in rodents, and it dramatically alters susceptibility to different carcinogens, and it alters it in two different ways. You can either increase the susceptibility to some carcinogens, and if it's gone, it decreases the susceptibility to others. So it's fascinating. It's not the kind of work I do, so this now gets handed over to our laboratory colleagues to try to explore what the mechanism might be, but it was a very interesting finding. But this did not tell the complete story about susceptibility for gastric cancer. So we've gone on and working with another group in China. We're trying to put together a very large set of subjects. We've used up to 10,000 people now to investigate other parts of the genome, and we're starting to build a panel of susceptibility alleles for gastric cancer in China. We're now taking this, and we're in the process of doing a similar study in people of European descent. So we can compare what it looks like in different ethnic groups. So I haven't touched on most of our work, but I did want to say that this is an important part of what we do, and it's important so that we can move forward with interventions. So now I want to turn to talking about opportunities for gastric cancer control. And first I'm going to talk about chemoprevention. Chemoprevention being the idea that you could deliver something in a pill form or something to reduce the risk of cancer in the future. In the 1980s, there was a lot of enthusiasm for nutritional interventions as a way to lower the risk of cancer at various sites, and the NCI sponsored some very large trials to look at whether or not nutritional interventions could be helpful. One of those interventions was called the Nutrition Intervention Trial, and it was conducted in Linshan, China. There was two different trials that in total enrolled 32,000 people, and you can see a picture of a group of people. These are basically all farmers waiting in line to go through their enrollment process of entering into this long-term trial. The general population trial just took adults 40 to 69. There was no risk stratification. There was no particular concerns. A few people were eliminated, people who had chronic diseases, but with the low level of medical care, you wouldn't have known who most of these people were who had chronic diseases. And using something called a fractional factorial trial design, the idea was to take 4 different intervention agents and give each of these intervention agents to half the people, and the other people did not have it. And there was a small group of people who had no intervention, and then it was about just over 5 years that they were given daily supplementations. So every month they were given a bottle of vitamins, and they were asked to take one each day, and they did this for over 5 years. And then they were followed closely for what happened to them. These were the intervention agents, the 4 different factors, and I've highlighted factor D, which was a mixture of selenium, beta-carotene, and vitamin E. The doses are about 1 or 2 times the recommended daily allowance, so these are not pharmaceutical doses. These are supplementation doses of these agents. And this was really a massive undertaking. This picture is of the empty pill bottles. So at the end of each month, when they got a new bottle of pills, the old one was taken back, and it was counted to see if they had taken all of their pills as a compliance measure, and then they had basically drifts of pill bottles that had to be picked up and put away. And so you can imagine to do this for 5 years was a huge undertaking. And this was during the days where China was much less developed than it is today. And the results were, in fact, quite exciting. The factor D significantly decreased total mortality, so mortality due to any cause, by 9%. There was a reduction in total cancer mortality, and then quite notably, there was a 21% reduction in gastric cancer mortality. I think that this remains the only trial of vitamins that has ever shown that they can do anything good for you. There's really no evidence that taking multivitamins does anything, and in fact, it may be unhealthy to do. But this was sort of stunning, and it was really exciting. And the problem is how do you translate this into public health action? Because first of all, there was 3 different agents, and we couldn't really be sure which agent did something good. And we couldn't do another trial where we just gave these 3 agents to see what would happen. You could imagine that was another millions of dollars in all those piles of empty bottles before you could answer that. So in an attempt to sort of take a shortcut, we did other things, and I just want to show you that this is the survival curves, and then I'll get back to the other story. So this shows the solid line is the people who didn't get factor D, and the dotted line are the ones who did. So about 2 years after they started taking these pills, there was a separation in the mortality due to gastric cancer, and that's when the trial ended. But we still follow these people today, and a few years ago we published a follow-up where we wanted to see if the effect was durable. And what happened is that effect was actually quite durable, because the red arrow shows you where they stopped receiving the vitamins. And yet the curves stayed separated. You might have imagined that losing the protection of the vitamins would cause the curves to snap back together. But that isn't what happened. So 10 years after they stopped taking the vitamins, the people who had been exposed for 5 years still had that substantial benefit and were at much lower risk of developing gastric cancer. We're about to publish another 10 years of follow-up, and we've actually seen that the durability is gone. It only lasted for 25 years, but that's not bad. So it's pretty interesting. But again, we didn't really know what it was, and we didn't think that you could take these kind of vitamin pills and deliver them to everybody in the high-risk region every year. We wanted to think of something more simple. And so we started doing what we call observational studies. We took the blood that we had collected from everybody at baseline, and we started measuring things, and the first things we measured were selenium, beta-carotene, and vitamin E. And in fact, the curves that you're seeing here show that the people who had the highest level of selenium in their blood before they received any vitamins were actually at the lowest risk for developing gastric cancer. But it wasn't all gastric cancer, it was only gastric cardiac cancer. So from these different experiments, we concluded that it was likely the selenium that was the beneficial agent, but we also had to conclude that it was only going to benefit cardiac cancer. So if you went to a part of China where they have mostly non-cardiac cancer, it probably wouldn't do any good. And we also concluded that we don't know this, but it probably only would really work in populations that are selenium deficient. Selenium deficiency is because of soil deficiency. It's not because of anything else. And so this exciting intervention probably only has a limited application. And when we wanted to try to do what we would call a demonstration project, where we deliver selenium to different parts of the high-risk area and not others and see what happened, it ran against the food adulteration scandals in China. So if we were going to put the selenium in the salt, and unfortunately that makes the salt yellow, and that made it really unpalatable to people, and they were very worried about having their food doctored. And so that was a no-go. And so currently we're trying to explore whether or not we could add selenium to the fertilizer, and so it would just get plowed into the ground, and we would slowly bring up the selenium levels in these areas and see what would happen. If we can do this, it'll be 10 or 20 years before we know whether it works, but if it does work, it works at about a cost of 1 cent a day per person. So again, it's a very long-term process, but we're hoping that we can find something beneficial for at least some populations. So nutritional chemoprevention, there's no other big trials going on, but my colleague in Iran is pursuing a different kind of chemoprevention. He was very interested in this concept of the polypill that was developed by Nick Wald. This was to prevent heart disease, and the idea was to give people a pill that contained a baby aspirin, a little bit of statin, a little bit of diuretic, and you would just give it to everybody at starting at age 50, and this would reduce their risk of heart disease. Well, aspirin in observational studies has been very consistently linked to having lower risk of gastric cancer. The same is true for statins. And so in Iran, there is a trial going on right now, a small trial where they're giving the polypill every day in a randomized placebo-controlled trial to reduce heart disease, and we're following the gastric cancer in this as well. It's too small to do a really strong trial, but if we get a small indication that it works, the plan is to go into a large trial of at least 30,000 people. Okay, so what else could we do? Helicobacter pylori eradication. So given that HP is the primary cause of gastric cancer, and that HP can be effectively treated with antibiotics, should we go out and try to completely eliminate HP from an entire population? And this is an idea that has taken hold. There's a lot of advocates. We even have some governments that are thinking that this might be the right thing to do. HP also causes ulcers, which can be a very important public health burden by itself. And so a couple years ago, IARC convened a workshop. The report was published in 2014 where this idea was considered. The conclusion was that it was too early to make general recommendations for population-wide programs to eradicate HP. So there have been a number of very promising trials. They have all been somewhat too small to be convincing, but there is growing evidence that taking people and using an course of antibiotics, eradicating HP, will lower their risk of developing gastric cancer. And now, part of the hesitancy, I think, from the workshop group was that there's a number of large trials ongoing. Korea is doing a definitive trial that they believe will determine whether or not Korea will start eradicating HP on a wholesale. There's a 200,000 person trial being accrued in China right now, and Japan is also doing trials, and in Taiwan. The benefits would be that you could reduce gastric cancer, and you would probably also reduce the number of ulcers, and so that would be wonderful, but there are risks. And essentially, if you give any kind of intervention that can help people, it almost by definition might hurt them as well. And the worries is one is you may increase antibiotic resistance. So there's lots of antibiotics being used right now, and in fact, one more course for a lot of people is sort of a drop in the bucket, but it all adds up, and you might end up with a lot more resistant bacteria, and not just HP. All the bacteria are going to be hit by this dose, and you could end up generating a lot more antibiotic-resistant bacteria. And the other thing, as I mentioned, is you might actually increase the risk of other diseases. Esophageal adenocarcinoma, it's possible that eradicating HP would lead to increased risk for this cancer. Now, all of the data about the protection against esophageal adenocarcinoma is probably from people who were never infected with HP, not adults who had their HP eradicated, so we don't really know. We also have to be worried that you're going to develop reflux disease once you no longer have the HP there. And there's not good evidence about this, but if that occurs, you can have a large number of people who have chronic illnesses. It's not as severe as cancer, of course, but if you flood a gastroenterologist's office with lots of people who are complaining about reflux disease, it could be a real risk, and reduce the cost-benefit analysis, or reduce the benefits. And then there are theories about the increase in asthma, obesity, that may be being driven by the lack of HP. And so there's a real need to understand all the risks and benefits before this is undertaken. But there's a lot of stuff that high-risk populations could do right now. Before these conclusions come, we need to know exactly what the cancer incidence rates are. So when we look in these countries, these low-resource countries in Central America that may not really know their gastric cancer rates, that's an important piece of the information. We need to know the prevalence of the HP infection. So in rapidly industrializing countries, HP is going away on its own. It may be that a lot of the young people in these middle-income countries aren't infected, and there's no point in contemplating eradication because maybe it's gone on its own and we don't really know what the rates are of infection. We have to have an eradication regimen that works in the country, specifically in that country, and we want to know what the reinfection rate is, because if you're going to get everybody reinfected right away, again, maybe it's not worth doing the eradication. So that comes to the last topic I wanted to touch on, and that's screening. So there are possibilities of reducing gastric cancer mortality by directly screening for the disease. Currently, Japan, South Korea, and China, and Taiwan actually are all performing some level of individual screening for gastric cancer, analogous to what we do in the United States for colorectal cancer. It used to be that they would use barium and X-rays to do this. Now it's mostly done using endoscopy. But there's some big challenges to doing it by endoscopy. We know that you can detect lesions. We know that once they're detected, you can remove preneoplastic lesions and you can remove early cancers with very conservative therapy. But you can imagine that the people who need this most are the least able to provide it. Going into central China and doing an endoscopy on every middle-aged adult is really daunting. You might have 50 million people that need to be screened. The Chinese government is funding 200 screening centers right now. They cannot screen 50 million people. We also know that in the stomach, you're stuck doing blind biopsies. We have no good way of highlighting where you're going to biopsy, and so the standard is to just take 5 biopsies from different regions of the stomach. There's a nice demonstration over here today that I saw from technology that may be able to improve that by pre-screening the areas that you're going to biopsy, but it's a difficult question. And of course, we have no idea what the recall interval is. And right now, there's no definitive trial evidence to support these activities. It's probably helpful to some individuals, but we don't know whether it really works as a medical program. We don't have definitive evidence from a trial that Pap smears work, but we know that they work. But the other side of the coin is PSA testing. We had to run a tremendous amount of activity around PSA testing with no proof, and now with our trial evidence suggesting that PSA maybe doesn't do any good as far as really truly reducing the burden of prostate cancer. So ideally, these programs get validated before they get into place, but in general, that doesn't seem to be the normal pattern of what happens. People get enthusiastic and they do these things, and maybe it's helpful, and we got to hope that it is. We're hoping right now, 2 weeks ago, I was in Turkey. Turkey is being actively considering doing gastric cancer screening. And what we're hoping is that if they do it, we can do it in a way that lets us test whether or not it's actually doing any good by doing it in a way of randomizing communities to being screened rather than just saying catch as catch can for a whole region. We know that for screening tests, we might be able to do better by doing risk stratification. Rather than just screening everybody when they turn 50, maybe we can pick out the people who need it the most. So we could build a risk model. We could do it using age and sex, family history, maybe a few other things. HP serology is appealing, but it only works in a place where you might not be infected. There's no point in central China of testing HP serology because everybody was infected, or 98% of them were. So what's the point? That wouldn't help. Okay. There's other ideas, and that's this other idea of a serological biopsy for gastric atrophy. So we know that pepsinogens are secreted into the blood, and that changes when people get an atrophic stomach and they're on their way to cancer. It has never been proven to work, but it's strongly associated with risk of cancer. Recently, our group has shown that ghrelin, which is the hunger hormone, is strongly predictive of gastric cancer risk, and we also have evidence that measuring vitamin B12 can be a strong predictor. And maybe if we take all of these different markers together, we can build a really good risk model and only screen, 10% of the population rather than 90% of the population. So I've summarized for you the worldwide burden of gastric cancer, which is high. The fact that our work suggests that there's some real etiologic differences between populations, and if we want to understand this disease and do something about it, we need to continue this work. There are at least 3 different good ideas about how we might control gastric cancer, but we don't have evidence right now that any of these should be employed in a population level. But the work is continuing. It's something that what we need is we need more people who are interested in this cancer, and we need more time spent on this cancer, and every cancer site feels that way. But when you have about 1 million cases a year occurring and you have a pretty small cadre of people who are doing this kind of research, it really cries out for more people getting involved. And I just wanted to finish by showing this picture again. I find this picture quite haunting. These people were quite willing to undertake a high burden on them. They were asked to come in and start taking these pills every day. These were Chinese farmers in the 1980s. They had a lot to do other than participate in research studies. And the way this woman was looking at the camera, it just really, I find it quite haunting, because we know that at least 3 or 4 of the people who were sitting here, on average, died from either gastric cancer or esophageal cancer while we were following them. And with that, I'll conclude. Thank you. So I'm happy to take a few questions if anybody has anything. Otherwise, I'll turn it back over to, yeah.
Are there any geological studies to correlate the selenium level in,
Yeah.
the area with the selenium level?
Yeah, so it's kind of stunning. If you look at a map of soil selenium in China, it's hauntingly familiar to the gastric cancer maps. Recently, one of my colleagues at IARC, which is part of the World Health Organization, who's doing esophageal cancer research in Tanzania and in Kenya, she showed me the selenium maps for Africa, and for at least for esophageal cancer, there's a lot of suggestion. And actually, somebody from my group is here today, Natalie, and she has a poster where we tried looking at selenium and esophageal preneoplasia in an African study. So, yeah, there's the geological evidence supports the idea. Yeah.
So you sort of left us hanging on the Japanese story. I wonder if you have any further thoughts on the paradoxical
The fact that they have such high rates of gastric cancer? Yeah, so that is a bit of a, I don't think anybody really knows the answer to why that is. There is some evidence that high salt intake is an important part of the gastric cancer risk process, and salt intake in Japan is pretty extraordinary. Their guidelines are, I believe the guideline is to try to limit yourself to just 9 grams a day. Whereas the target in the US is 2 grams a day. And so their rate, their salt intake is really extraordinary. Other than that, I don't think that we really know what's going on. But they do have, I can't say that the screening program is highly effective, but if you're an individual and you get gastric cancer screening endoscopy in Japan, they're very good at it, and they can find a lot of small lesions, and they can remove those lesions with endoscopic surgery, and it's essentially you just go home and you're fine. You keep coming back in to get screened, and so it's interesting. I just don't know that there's any, they haven't really built the right kind of evidence by doing a cancer control trial, but that's an extraordinarily big undertaking to do that. I don't think there's any evidence that it's particularly a genetic thing. I think it's much more likely that it's something else about the environment that we don't understand. Yeah.
One more question.
Sure.
Besides the HP, have there been any studies on the microbiota of the cancer incidence?
Yeah, so that's a really good question. There's a small subset of gastric cancers that are linked to Epstein-Barr virus. About 5% of all gastric cancers are EBV positive. It's not really clear how that works. There was just this recently, a big sequencing study looking at the mutation profile, and the people who have EBV positive gastric tumors, the mutation profile is quite different from people who don't have EBV. But looking at other bacteria, that's a major part of my research program now. And I've got very interested in this. I've done a lot of studies on how poor oral health increases the risk of these cancers and the link between periodontal disease and these cancers. And with the new DNA sequencing technology, it's really ripe for interesting studies at looking at how the bacteria in the mouth, the esophagus, and the stomach are interacting to metabolize food and other carcinogens and interact with the immune system. But right now, we don't have any clear, there's nothing that we can really say about that. The technology is exciting, but it's been really poorly implemented, and so we've spent the last 2 years developing highly good quality ways of doing this sort of analysis to rapidly characterize the entire bacterial. That doesn't get at all the viruses, it doesn't get all the fungus, it doesn't get everything else, but we're working on the bacteria. And in fact, we are planning, with our colleagues in China, we're just about to start a new program. We're going to collect about 1,000 people with a range of everything from just an HP infection to different grades of preneoplasia up through early cancers, collect their saliva, the gastric juice, the stool, biopsies and brushings, and then characterize the bacteria across that spectrum so that we can try to sort of get a handle on what role that might play either in the causing the disease or possibly helping us predict who's got the worst lesion, where we could do something simple. So we do have a system of taking a sponge that's inside a capsule on a string, people swallow it, the capsule dissolves in the stomach, and we pull the sponge back up, and we've then sampled the esophagus and the stomach. if we can start to characterize things in there, including the bacteria, that might be useful. Yeah.
And thank you for an excellent presentation. For the risk prediction and stratification model, could you comment on whether it makes sense to include chronic gastritis or individuals with a history of chronic gastritis, especially atrophic gastritis?
Yeah, so that's probably, I mean, the people who are on the path towards cancer have chronic atrophic gastritis. The atrophy means that they've stopped secreting gastric acid and puts them at higher risk for gastric cancer. The number of people who know that they have it is very small, and so it's something that you probably have to go out and measure. And that's where this idea of measuring pepsinogens, maybe ghrelin, vitamin B12, all of these are serologic markers that we could get at whether or not they have atrophy without actually having to stick something in their stomach to do it. But we think that if we're going to build a risk model, that probably would be part of it. Yeah. Yeah. Yeah, so the question was about smoking. So smoking is a very important risk factor for these diseases. For gastric cancer, it's very consistently about twofold increased risk. The smoking rates in that part of China in 1980 were actually pretty low because the farmers don't have any cash and they can't buy tobacco. And so there wasn't much smoking. The women essentially don't smoke at all. Most of the men, majority of the men smoke, but they only smoke a modest amount. And that's actually with the increasing, as the economy rises in China, it's probably going to become more of a problem because they have more cash. In the trial, since it was a randomized trial, we didn't even have to control for smoking. In our cohort analyses, we control for smoking, but the association is quite modest for these places because the total exposure is low. Yeah.
Bob Hyatt again from UCSF. Do I remember correctly that back in the day when Julie Parsonet from Stanford was working on this, that she founded an association with gastric lymphoma?
Yeah.
What's ever happened with that?
Yeah, so I think it seems that there's a specific type of gastric lymphoma that is caused by Helicobacter pylori, and that at the early stages you can actually treat the cancer by treating the bacteria and remove it. But that is such a small part of the gastric cancer burden that it hasn't really, there isn't really that much research on it because you have to assemble huge numbers of patients to find enough lymphomas to do any work on it. But I think that is, if somebody is fortunate enough to have an early stage gastric lymphoma, it may be that the antibiotics would be enough to treat the cancer.